Okay, excellent. Thank you for coming, everyone. I looked through the invite list or the registrant list, and nice to see some familiar faces and some folks who've helped on this project. So we're going to talk about Western hognose snakes in Texas. You know, how many species are there and where do they still occur? I want to say that it's been a dream to work on hognose snakes. I have been fascinated with them since I was a small child. They're one of my favorite organisms just overall. I still have some at home, right? I enjoy keeping them. I enjoy finding them in the field, and it's been just a great pleasure to work on them and work on them in the state of Texas, where they're very, very interesting animals. Okay, so a little background. Heterodon nasicus, the Western hognose snake. It's a small to medium-sized snake. It eats rodents, lizards, and amphibians. We'll talk about, what they eat and why in a little bit. They're fossorial, so they spend a great deal of time underground. A majority of their lives are probably spent underground, and a lot of the things that they eat spend a lot of their time underground. And a lot of people know this. The most interesting thing about a hognose snake is that they do death feigning behavior. So if you come across a hognose snake like that photo there on the left, and you look down and you can see that animal there is already reacting to me taking its photo, and it's flattening its head and it's trying to look threatening, probably to look venomous, and it's starting to curl up a little bit. And I found that animal was stretched straight out. It was traveling across a road and it's trying to look threatening. And if you go pick that snake up, it's not going to bite you. It may strike at you. If it strikes at you, it's going to strike at you with a closed mouth. Very occasionally, a hognose snake will bite in defense, but it's very, very rare. They almost never bite in self-defense. But pretty quickly, a wild hognose is going to go into death feigning behavior, and it's going to flip over and roll over and start writhing around. And if you give it a couple minutes, it's going to stop and it's going to stay upside down. If you put that snake right side up, it will roll back over on its back an unlimited number of times. It can only be dead to the snake if it's on its back. They will musk all over you. Sometimes they will regurgitate prey. But this is really, really interesting behavior. And all the northern snakes do this. So, and if you go grab a wild hognose, there's a 99% chance that it's going to do this cool death feigning behavior. So in Texas, we also have Eastern hognose. We're not going to talk about them very much. Eastern hognose are distributed broadly across Eastern North America. They go all the way to the hill country of Texas and then the panhandle. They're not in far west Texas. They obligately feed on amphibians. So a Western hognose, a lot of times will gladly eat a lizard or in some cases a rodent. But an Eastern hognose without really, really significant sort of scenting in a captive situation using other things to scent prey items there's probably no Eastern hognose that are grabbing rodents in the wild. They are obligately amphibian feeders. They're very difficult to keep in captivity for that reason, and many people have struggled for many years. They are even better at the death feigning behavior than a Western hognose, though, and they're incredibly dramatic. So that being said, I still think they're way less cool than a Western hognose. That's just my opinion, but that's what you get. Okay, so a little trivia about hognose. They are venomous. We as humans tend to think of venom as a medical definition, but people who study toxins would tell you that numerous snakes are venomous, things that we would consider classically non-venomous, and some lizards and various other things as well. That right there is my hand. That is about 48 hours after I was bitten by a captive bred Western hognose snake. And I just told you they don't bite, right? Well, especially in captivity, they are voracious feeders, absolutely voracious. If you get them going, they eat really well. And I picked this snake up to clean its cage, and it latched onto my finger, thinking that my finger was a prey item. I probably smelled like mice. And that snake chewed. I didn't want to rip the snake off. And so I finished cleaning the cage and I let that snake chew. It was attached to my hand for more than a minute, but probably not 2 minutes. And I pulled the snake off and cleaned up the wound, and everything sort of seemed fine. And it was never really truly painful, but it started to swell. And 6 hours after I was bitten, the volume of that finger was approximately twice the volume it should have been. And it got very stiff. And then I started getting some light tissue necrosis. So they are venomous. We're very lucky in terms of captive snakes that they pretty much don't ever bite except in feeding situations. But even then, if you're smart, you can avoid that. So I will likely never let a hognose snake chew on me again for 1 to 2 minutes. But to be fair, I've been dealing with them for multiple decades, and that's never happened to me before. But kind of interesting. Other interesting things. They are probably mimicking rattlesnakes. Now, mimicry in a scientific sense is very difficult to prove, especially in things that are cryptic, right, that are trying to hide. It's a little easier to sort of get good evidence for mimicry in things that are aposematic. Think coral snakes and milk snakes. That's a little bit easier to get information on. But are these snakes converging in color and pattern because that's a really excellent color and pattern to hide, to not be seen by predators? Or are they converging in phenotype, in color and pattern, because the hognose snake is trying to mimic the rattlesnake? So I'll let you decide which is which, but I'm going to tell you. The one on the left is the hognose, and the one on the right is a Massasauga rattlesnake. Now, you can see they're reversed in that photo. I took that photo with a white piece of poster board, and I put a pane of glass over the snakes, and that way I could take this photo safely. You can see the glare there, which I couldn't eliminate. And you can see how close their color and patterns match. And I take this very seriously when I'm in the field. There have been several times that I've looked down and seen just a chunk of snake, just an inch or two of snake crawling through vegetation. And I don't grab the snake because I'm not sure if it's a hognose or if it's a Massasauga rattlesnake. They may also be mimicking other rattlesnakes as well, but their closest mimic is almost certainly the Massasauga. They have some other behavioral things that they do. That tail curling there that you can see in the hognose, some people have hypothesized to kind of look a little bit like a rattle. There's really no evidence for that. And Heterodon so hognose snakes do some head twitches sometimes. There's also thoughts that sometimes Heterodon come up off the ground and hiss a little bit, and they flatten that neck out, right? That's the joke, is that they're the spooky cobra. And they flatten that neck out, and they tend to do this, come off the ground more in places that there are things like prairie rattlesnakes, which do that kind of classic rattlesnake high guard position. So they're probably mimicking rattlesnakes. Regardless, they are very, very cool animals. And, you know, for a snake that's mostly brown, they're surprisingly beautiful, right? Okay, so kind of the meat of the talk. Why do we care at all about how many species they are? Like, why is that even a question? Okay, so species are the currency of how we talk about biodiversity. So if we don't have an accurate accounting of biodiversity, we can't do all sorts of things. We can't talk about organisms in a distinct way, right? That would be the equivalent of looking up and saying, looking at a condor flying by and just say, well, that's a bird. And it's the same as a sparrow in the McDonald's parking lot, right? So it gives us an accurate accounting of biodiversity. This increases our understanding of the organisms around us. Sometimes this has very functional benefits to humans, right? Certain species are very useful to us as humans for all sorts of things, food, medicine, clothing. Lots of species are not really that useful to us. Okay, so sometimes there's very functional benefits. From a biodiversity standpoint, it helps us know where to apply our conservation efforts. I don't have to tell anyone that there is both limited money and limited time and effort. We only have so much money that we can distribute to threatened and endangered species, and there's only so many people who can work on them, and that's a function of money as well. So I want to point out something that this gets a little contentious at times, but species are real entities in nature. You can think of them as natural kinds, a kind of thing. and they're diagnosably distinct from other kinds. So people will say taxonomy is all made up, right? That it's all just humans putting things in one box or another. And it's not. The labels that we use are created by man, but the entities exist in nature, right? You can go out and you can tell one entity from another. Now, that doesn't mean that it's easy, and that doesn't mean that sometimes the boundaries aren't fuzzy or that the boundaries are ever changing. But those entities do exist in nature. Okay, so we wanted to know how many species of Western hognose snake there are in Texas and range wide. And you're going to see in a second, most of the interesting stuff about hognose, Western hognose, is taxonomically is going on in Texas. So here's the historic distribution of hognose snakes. This is Texas pulled out, and these are all the museum records in the last 40 years. You can see in the little insert on the top right, you'll see the total range of the Western hognose snake. You will see some little disjunct populations in Illinois, Iowa, Minnesota, even Missouri. We'll talk about that one in just a little bit. And it goes all the way north into Canada. It goes far south into basically the top of central Mexico, and west into Arizona. And the map goes a little bit farther there. So these are the records within the last 40 years. Now, you'll see, pay attention to those red dots on the right. Those are what people used to call the dusty hognose, Heterodon nasicus gloidii. Okay, so I will tell you right now, spoiler alert, those animals are gone. There has not been one found, a reliable record in Texas since 1996. They are gone. Is it possible they still exist? Yes, but they're almost certainly gone. And some of you who are really knowledgeable might disagree with where the blue and the yellow is. And that is a complicated question. And that complicated question goes up into New Mexico a little bit too. But you can see there's a line there. That crooked line, if you can see right here, this is the Pecos River. And the Pecos River is generally considered to be the dividing line between Western and Mexican hognose snakes. Okay, so the Western hognose would be Heterodon nasicus nasicus, and the Mexican hognose would be Heterodon nasicus kennerlii. Talk about this. Okay, so what's the taxonomic history? This is where it starts to get muddy. Heterodon nasicus, the true Western hognose, is described by Barrett and Gerard in 1853 from Amarillo. Heterodon kennerlii is described by Kennicott in 1860 from South Texas. Talk about this. For more than 100 years, people are arguing as to whether they are separate species or not. There's no doubt that they are super closely related. They may be the same thing, but people are arguing about this. And there's, to be honest, there's a herper component too. People who like reptiles and amphibians as hobbyists, because people love hognose snakes, right? People think these are cool, fun animals to find in the wild, which they are. They're charismatic animals to hold and to look at, and they're enjoyable animals to keep in captivity, which is a very popular thing. So Kurt Eckermann comes along and over several papers, 1996, 99, 2003, looks at essentially all the hognose snakes he can find in museums. And he does not do any genetics. This time period predates a lot of what we would call modern molecular systematics. And he does scale counts. And I'm going to spare you all the gory details because he looked at everything, tons and tons of stuff. And fundamentally, all he could pull out was this one character. And that character is an azygous scale count. So you say, what is an azygous scale count? Okay, if you look at the big rostral scale and you come back towards the eyes, but not all the way to the eyes, I don't know whether you can see my cursor, but in front of the eyes, but behind the nose, there's all these small scales. And so Kurt Eckermann looked at basically every hognose snake, Western hognose, that you could get in collections. And when he looked at them, he could only pull out this azygous scale count. That's all he could pull out. And what he noticed was animals to the north tended to have way more azygous scales than animals to the south. And he was able to split it into two groups. He was able to split it into 6 and fewer azygous scales and 9 or more. And you will get azygous scale counts into the teens or even the 20s. So a lot of azygous scales on like a Western Hognose snake from Colorado or something. And so Eckermann could only pull out this one characteristic that was really distinctive. Now, I will tell you that blotch count goes up in the east and goes down in the east. I said that backwards. Down in the east, they tend to have fewer blotches in the east and they have more blotches in the west. But that's not even a hard and fast rule by any means. So what Eckermann found, though, was that 97% of these animals could be put into one of these two categories. But that being said, there were several animals, quite a few, that he couldn't place, that were between, basically had 7 or 8 azygous scales. And that was fairly common.
Yeah, the link came through, but it's because I think it's sharing an internal like UTA thing. It won't let me get it.
Okay. Well, let me try one more time and I'll keep talking.
You can't download the file and send it. That's too big.
No, it's not seeming to work. I don't understand why WebEx is not even letting me, at this point, share my screen.
Yeah, it's weird as it worked earlier.
Yeah, and it works twice.
You're on a Mac, you said, right?
Correct. They wreck everything. Okay, let's try it now.
I see your screen.
You see it?
Yeah.
You see the presentation?
Yep.
Perfect. Okay, good. So 97% of these 350 animals fell into one of these categories. Now, what I want you to notice, if you look over here at the map and you look at the cursor, you will notice that there's this dot right there. And the star above these two dots, that star is the type locality for what they called Heterodon kennerlii, the Mexican hognose snake. That's the type locality. And I want you to notice something. It's in the shaded area for Western hognose snakes. And Eckermann just drew a line. He didn't even suggest a barrier, right? He said, well, here's the line, and animals below this line almost always fall into this category, and animals above this line fall into this category. And there's several people who've noticed that there are interesting hognose snakes in these areas that have weird azygous scale counts. The same thing happens over here between essentially the Waco Mountains and the Guadalupe Mountains. You get weird hognose snakes. So Eckermann didn't suggest a barrier, and he didn't have what we call a fixed morphological difference. He couldn't tell anything that said we can always put these snakes in one category or the other. And Eckermann said, well, I'm going to make them two distinct species, and we're just going to say that there's hybrids. And that's a valid viewpoint, right? That there's hybrids. So, but now we bring in genetics. I'm not going to bore you with a ton of nuts and bolts on this. We generally consider this at this point to be more reliable than morphology, because in theory, you're counting at this point millions of base pairs, millions of potential characters. So that scale count is one character you can count. It's one trait. You can count millions of traits. It can be what we call neutral variation. The number of blotches a snake has is highly influenced by natural selection. A lot of DNA, a lot of the genetics are less influenced by natural selection, at least in theory. Now we can sequence entire genomes. The hognose genome is in prep. We can now compare a snapshot of the entire genome using this thing called dd-RADseq. And essentially we get a snapshot of the genome, and it's the same snapshot for every individual. So you're comparing apples to apples. Okay, and I will tell you right now, the most difficult part of working on hognose snakes is collecting high quality DNA samples. We have over 150 samples from the entire extant range, all the way from the Canadian border, all the way down through Mexico. And let me just tell you that 150 doesn't sound like a lot, but with hognose snakes, that's a lot. They are difficult to collect. Most people have not seen that many in the wild. Okay, so let's just jump right into it. I'll tell you right now, these are whole mitochondrial genomes. So this is not DD-RADseq, and each one of these individuals, each one of these numbers represents an individual hognose snake. And the first thing you will say is, wow, there's a little bit of separation there. But if you look at the scale bar down here, it's 0.1 substitutions per site. This works out to a percentage at this point. This is not even 1.5% difference. So that's the first thing, okay? The second thing I want you to notice is that if you try and classify these animals by where they're supposed to be from based on Eckermann's classification, you get multiple Western hognose that have mitochondrial genomes much more similar to Mexican hognose. You're like, okay, so that's weird. That's one problem. You don't have monophyletic DNA. You don't have DNA that you can separate out and say, this is always this way, and these are always each other's closest relatives. You just don't. There's another problem if you know anything about phylogenetics, and that other problem is this entire Kennerlii clade is nested within a subclade of these nasicus. This means that these animals down here are more closely related to Mexican hognose than they are to any of these animals up here. In the same way that you and your siblings are more closely related to each other than either of you are to your cousins, because this group is nested within this, this makes what we call classic Western hognose paraphyletic with regards to Mexican hognose. So there's no good phylogenetic justification for calling them two different species. And there's multiple reasons for that. We'll talk about this. Now, if you look at this, this is a kind of cool thing from the RADseq, and you say, all right, each one of these lines is an individual hognose snake, and you can try and put a label on it and classify its genes. If you tell the analysis, this is for the RADseq stuff, so the entire genome, you can tell the analysis to put it into two populations. It can tell you the red in this case is nasicus and the blue is Kennerlii. First thing it'll tell you is Gloidii just groups out what we used to call classic dusty hognose with nasicus. So nothing crazy there. Kennerlii, there's some individuals that are pure Kennerlii, but there's some nasicus that group as Kennerlii. These animals are exchanging genes across their contact zone. If you try and say, hey, make sure there's three populations, to pretend Gloidii is real and fit these animals genotypically into one of three categories, it can't do it. It says, man, we can't even get anywhere with the Gloidii. And then the nasicus are going to have a bunch of Kennerlii genes in them. Okay, so look at this. But notice that the Kennerlii in both analyses, the Mexican hognose, always comes out as monophyletic. And you can see that, right? This Kennerlii clade here, there's no blue up here in the red, but there's red down here in the blue. Okay. All right, I mentioned something before. Dusty hognose, what we used to call Gloidii, they're extirpated in Texas. We don't have high quality DNA for them. There's one remaining population in Missouri, and we were fortunate to get tissue samples from 2 live animals from that and plug them in. And they just group in with classic Western hognose nasicus. Okay, so how did we get these two forms? Like, an experienced herper can look at them and say, well, that's a Mexican hognose on the right, and that's a plains or true Western hognose there on the left. And you can say, well, this animal is bigger. It has a lot more blotches. A lot more of the blotches are double blotches. Okay, and if you really dig in there, it has way more azygous scales. Okay, this animal has fewer azygous scales, fewer blotches, right? Clearly, there's some structure here. They do look different. Well, how do we get new species? This is kind of your classic speciation biology. You've got an original population. You get a geographic barrier that forms between them. They begin to evolve differences in isolation. And if they evolve enough differences, they will become separate species. And even if they come back into what we call secondary contact and they come back together, at that point, the barrier is so good that they won't breed. So We believe this is the mechanism by how most animals speciate. How we get most species of animals is through this classic allopatric speciation. So I want you to look, and this is an iNaturalist map of hognose observations, current hognose observations. You can see some of these animals. There's the Missouri population here, some animals in Illinois. Don't worry about these ones over here in LA and Ohio and Virginia. We'll come back to those. They go up into Canada, and then you can see the classic distribution for what we call Mexican hognose, right? And this is reliant on people to come in and ID these and put these in the correct categories. And then I want you to look at this map, this temperature map of the United States. And obviously South Texas is really, really warm, right? Warm like peninsular Florida, very, very warm. Warm like southern Arizona, okay? Very warm place, obviously. And you say, well, what caused this? This is 17,000 years ago. And this is what we call the last glacial maximum. And you can see the extent of the glaciers in North America and how far they come down and the temperature derivation from where we are today. So much, much, much colder. Okay. Now I want you to look at this little finger that extends up here, potentially into what we would be Colorado even now. So there are places that aren't too terribly cold, right? And what very likely happened is these hognose snakes died out where the glacier came and made it too cold, or even where it wasn't glaciated, it was too cold for them to survive. But animals survived in Mexico and probably South Texas. And then, but there were probably occasionally populations up here in these little warm patches that made it. Okay. And they started to develop differences. And you say, well, what causes this cycle? How do we get glaciation and stuff? And it's precession. So everybody knows that the Earth rotates on its axis and it revolves around the sun. But probably the next most important motion that the Earth does is precession. It wobbles. Anything that spins wobbles. And so the wobble cycle takes about 25,000 years. And when you are wobbled sort of straight up and down, your seasons are less pronounced. And when you're wobbled at a wide angle, your seasons are more pronounced. And since they're so much more pronounced, this allows glaciers to build up in the winter. Okay. So this Mexican refugium pattern, where we get these animals pushed down into Mexico and they recolonize north, is repeated over and over and over. Toads, earless lizards, ground snakes, coral snakes, a couple different species of rat snakes, black-headed snakes. These are some animals that we've worked on called Sonora ground snakes. And you can see there is essentially a break here between these orange dots and these yellow dots. And this is a case where the biogeographic barrier, the allopatric speciation, went to completion and actually created a barrier that when these animals came back into secondary contact, they couldn't breed. It didn't happen with hognose snakes. Talk about that. This is one that we've also worked on. These are the earless lizards, and this is a map from Ralph Axtell's monograph showing you earless lizard distributions. And you can see there's this disjunct in the range between now what we call Holbroochia subcaudalis down here in South Texas, and to the north, Holbroochia lacerata. So lots of animals follow this pattern. And the Balcones escarpment in Texas is generally considered to be the barrier. Sometimes you'll get the Pecos River if the organisms continue west. But this pattern is repeated over and over again because this glacial cycle is repeated over and over again. And how far you get north, like hognose snakes made it all the way to Canada. It's because a hognose snake during the course of its life can crawl several miles. So as the glacier began to recede 17,000 years ago, you can see that animals followed it northward as the climate warmed. So how do we get these two different forms of hognose snakes? Two possibilities. The most likely is that allopatric speciation went, but it didn't run to completion. You didn't get reproductive isolation. So when these animals came back into contact, they bred. It's also possible that there was one population of hognose snakes, and as it moved northward, it developed local adaptation and change. That's less likely. Okay, we think that there was probably a population of animals somewhere in the Central Plains that did survive, but they didn't survive in the area between Mexico and South Texas and the north. And when they came back into contact, they developed differences, but not enough to keep them reproductively isolated. Okay, so conclusions. There's one species of Western hognose in Texas. It's Heterodon nasicus. There isn't reproductive isolation between the populations. There's no fixed morphological character difference, which is your generalized standard for morphological systematics, a true fixed character difference that always holds true. There's no molecular synapomorphies. If we go from the early days of molecular systematics, the old adage was that 2% divergent in mitochondrial DNA. They don't even meet that. There's no reason, there's no molecular synapomorphy to say we can always tell them apart. And then most importantly, from a phylogenetic sense, they are very paraphyletic, right? If you're going to call the two species, if you're going to call the two Western hognose snakes two species, then that's the equivalent of saying that birds aren't dinosaurs, right? Birds are dinosaurs. Everyone accepts that now. They are reptiles. Obviously, birds are reptiles. Well, one clade of hognose snakes is nested within the other because one of those is an offshoot of the other as they shot north. So there is some noticeable genetic structure. Like, we can look at this and we can look at the animals and tell them apart. So if you really still believe in the concept of subspecies, you could say there's two valid subspecies. You could say Heterodon nasicus is the plains hognose, and Heterodon nasicus canerlii is the Mexican hognose. The dusty hognose is not a valid taxon under any definition. This is the big thing. All animals essentially north of Texas, and even probably in the Panhandle, represent recent recolonization after glaciers begin to recede 17,000 years ago, which is the last glacial maximum. So from a conservation standpoint, hognose snakes have declined. They've been locally extirpated in many places. There's no records in East Texas for the last 25 years. The old thing we called the dusty hognose of East Texas is gone, but it never really was distinct. The last good record from Oklahoma is about 2007, and there's still that Missouri population, but they are just Western hognose. They've had tremendous declines in South Texas. Probably from invasive grasses. If you can look at this grass here, this is buffle grass, which is a grass that was planted all over South Texas for livestock forage. And unfortunately, it creates a different spatial type of ecosystem. Hognose snakes, like a lot of South Texas ground fauna, want to move through bunch grasses where they can move along in areas of bare soil between the bunches of grass, and buffle grass can create an impenetrable carpet. And it turns into a biodiversity desert in some places where it doesn't allow a thriving diverse ecosystem. Amphibian declines have probably impacted them because they eat them. And then fire ants cause a huge problem for all egg-laying reptiles. Fire ants will eat anything. And their decline is certainly multifactorial, but these are some of probably the root causes. I will tell you that collection for the pet trade is most likely not impacting wild populations, and here's why. They're incredibly difficult to find. It is so difficult to find a Western hognose, even if you know what you're doing. You have to go out at the right time, spend hundreds of dollars in gas money. They're tough to find. And most importantly, at this point, wild phenotypes are less desirable. Hognose snakes, I'm going to show you in a second, are interesting. And if we look at this historic distribution again, I just want to tell you that, okay, well, the red stuff is all gone at this point, right? And now we could just draw that line wherever you want. We can acknowledge that this boundary here is fuzzy, and people were acknowledging that in the 1990s, and people were acknowledging that in the 1800s, and we're still there with knowing that that boundary is fuzzy. Okay, this is fun. Here's our current recent distribution. Now, I told you don't look at these other ones. So there's the classic core range of the Western hognose. Obviously, it goes down into Mexico. Look at this. There's a dot in Los Angeles. There's a dot in Ohio. There's a dot in Virginia. And then there's a dot somewhere over here on the eastern seaboard. And these are introductions. There at this point are tens of thousands of hognose snakes that are produced in captivity. They lay large clutches of eggs. These are all animals that you could not find in the wild in any way, shape, or form. They have about as much in common with a wild hognose snake as a husky does to a gray wolf. And people are keeping them literally by the tens of thousands. And unfortunately, for the animals themselves, but also for us, these animals get loose, and people find them and say, hey, I found a hognose snake in my local park. They are arguably one of the top 5 pet snakes in North America. And they are just a really charismatic animal that people like. I wish they wouldn't lose them and release them, but they are a charismatic animal. So I want to thank a whole bunch of people for this funding and support. Parks and Wildlife and Fish and Wildlife Service. I've been working with my collaborator, Nathan Rains, for a long, long time, and that's been a lot of fun and very productive. And thank you to Paul for all his support and what he's helped us do. Here's the big one, tissue donors. We would never have been able to do this study without literally dozens of tissue donors, some of who are listening to this right now. So thank you. Hognose snakes are incredibly difficult to collect in numbers, especially from specific places. And we really appreciate everybody who gave us tissues. My collaborator, who I've worked with for many years, Dr. Matt Fajita, if you want to talk to an actual real smart person, go talk to Matt. He loves to look at the data and do lots of fun computer programming to run these analyses for us. So numerous students, Dan Dudek and TJ Fernino, did a lot of work on this. And then I need to thank Troy Hibbets for his photography and a lot of his expertise over the years. He lets me use a lot of his photos for presentations because I'm not a good photographer. So I'd like to thank everybody. The herping community has been tremendously generous with this, and we couldn't have done it without their help. So with that, I will stop sharing my screen. I'm glad it survived all the way through, and I will take any questions.
Yeah, good stuff, Corey. Thank you so much. Yeah, and just as a reminder, pop your questions in the Q&A and we can read them out. I'll read them out to Corey and get him to answer them for you. There's a few initial questions, Corey, about your bite, as you might imagine. So we'll get through those first. Yeah. That's a good warm up there. So any other physical effects to the venom besides the sausage finger?
No, swelling and tissue necrosis and a little bit of pain, but mostly associated with the swelling, but the pain really wasn't bad at all.
You did say you probably wouldn't let yourself get bit by it again.
No, because I couldn't use my finger for 3 days.
Okay. Well, probably was the key word there, I thought. Okay, so how long did it take to heal?
It was back to completely normal with no signs of anything in about 2 weeks. It was functional in about 4 days.
Okay. There's a question about hognoses in captivity. I think, whether you can keep them in captivity if they're wildlife, there's a different definition. I'm sure the states all have different rules throughout the range.
They are legally collectible in many states that they occur, but I will tell you that 99.99% of all the hognose kept in captivity at this point are captive bred, and they are designer hognose. So, like I said, they have about as much relation to a wild hognose snake as, I don't know, a dairy cow does to a free range cow, right? They're very different animals.
Okay, somebody asks here, could Corey's reaction to the hognose snake bite have been caused by bacteria rather than venom?
Possibly. I've never been envenomated by any other snake, and it certainly presented all the symptoms of envenomation. I was getting swelling within an hour.
There's a follow-up said, everything I've read about these snakes says they are not venomous and that they do not have venom glands.
They've got venom. I'm not a toxicologist, nor am I a venom researcher, but I would encourage you to go read up on that. There's a lot of fairly new, last 20 years primary literature on lots of snakes that we would classically call non-venomous being venomous. Now, that doesn't mean they're medically venomous to humans, but are they producing secretions that they're using on their prey that are helping them overpower or even digest prey? Then absolutely.
Yeah, I think that's an important point. Venomous snakes get a bad rap a lot of the time, don't they? So we don't want to expand the list of snakes that would be on that medically venomous.
Absolutely. They're not medically So,
Yeah. Okay, this is a good question. Something I was curious about too. So this question was before you explained your hypothesis for the population genetic patterns. It says, so does Kennelii being nested within nasicus imply southward range expansion from a north
yeah, that's a.
refugium?
I knew someone would catch that. That's a fun artifact of the way that particular phylogeny is presented. Right. What's happening there is that I'll just tell you right now, and this we see this pattern all over the place. This is like an expert level question, so thank you. There's so much more genetic diversity from all these animals, regardless of what they are in the South, because they were there longer and they existed in larger population sizes. The stuff to the north are all these colonizer genotypes that are all these population bottlenecks. So there's way more genetic variation in the South than there is in the North. But simply because they're older lineages.
Got it. That's great. You touched on, actually, let's stick on the genetics for a second here. There's a comment here, there's a question. This is going to take me a while to read out. It says, very nice analysis, but I would say subspecies are not a matter of belief. It's just a way of recognizing different geographically distinct regions within a species, which is a factual observation, blah, blah, blah. One may choose to recognize distinct populations or not, but it is not a question of belief. It is just a question of whether we wish to recognize phenotypic differences within species taxonomically or not. You would agree with that statement. Yeah.
Yeah.
Very good. Okay. So what you mentioned the impacts of populations in South Texas. Somebody asked a question about the role of the energy extraction, fracking, oil infrastructure, things like that.
It's hard to tell. It would be almost impossible to design an ecological study where you would look at hognose snake populations because they're just so enormously difficult to find. Even in the classic way that you would try and get a decent number of them on a landscape would be pitfall trapping, and anyone who's ever pitfall trapped in hognose snake territory knows that they are not a common catch.
Yeah, very good. And that's a follow-up question here. It says, knowing that Western hognose is difficult to find in the wild, how do we identify and quantify a population decline?
I will tell you, it's all anecdotal. Literally, there's no, I mean, you can say that these animals were at this site and now they're not. But even that, I mean, these animals pop up years later where they haven't been seen in a long period of time. I will tell you this from a recreational herping perspective. So going to look for reptiles and amphibians, people used to talk about going to South Texas to go herping, and they would expect in maybe the course of a weekend or a week-long trip to find a hognose snake. Or if they lived down there, they would find several a year. And now finding a hognose snake in South Texas is cause for celebration. It's a rarity, and people who live in their range, who know what they're looking for, can't find them.
It's remarkable. Was the
phylogeny better northwestern South Texas? You get towards Del Rio a little bit, and it's better. But like some of the classic areas in true South Texas, they're just mostly gone, or they're in such depressed densities we can't find them very easily.
Was the phylogenetic tree you showed rooted with Eastern hognoses?
Oh, excellent question. We do have trees that are rooted with Eastern hognose. If you want to know that, we know that there's a third species that doesn't occur anywhere near Texas, Southern hognose. And Southern hognose and Western hognose are sister species. And then Eastern is the third one. So yeah, it's rooted with that. You're going to laugh. We accidentally, this is just kind of fun for you molecular people. We mixed up some samples one day that we thought were hognose, and we had to run a bunch of analyses over several times because we accidentally tossed some earth snakes in there.
Oh, wow.
So we got some good rooting with earth snakes. Accidentally.
Okay. So what do you think these snakes do during the winter freezing weather?
So they just go underground and sleep, and classically brumate like all reptiles in there. I think, during glacial cycles, a lot of these animals just died out. They weren't migrating away from the glacier. They just couldn't survive any longer.
Can you suggest any other resources where people can read about the Mexican glacial refugia situation?
I can, but they're largely in like Journal of Biogeography type journals, biogeographic journals. This is a common pattern. Another one of my collaborators, Christian Cox, he has numerous papers on and I'm on some of these as an author. He has numerous papers on a lot of these plains animals that, you know, move northward, and you see their genetic diversity decrease as you go farther north. So this is pretty well known in plants, anything that moves slowly. Gophers. Sam Kieschnick, who works for Parks and Wildlife, worked on gophers with this issue.
Oh, very interesting. There's a few more questions here. So do you think that you had mentioned this? Do you think the decline in hognose is related to the decline in bufo, in the toads?
It's certainly possible. It's hard to tell, but amphibians have notably declined in many places, and things that eat amphibians are obviously going to follow that.
There's a few reports here of people seeing hognoses further east and north in Texas. Somebody mentions one observed in Leon County that they suspect was an eastern hognose sightings around a neighborhood in Georgetown along the San Gabriel River.
And those are generally eastern hognose. One of the issues with getting Western hognose samples is that if you ask the general public for help, even the educated herping public, people will give you eastern hognose like crazy. And eastern hognose have declined too, but eastern hognose are still common across the majority of their range in Texas. So, or at least occupy a majority of their historic range in Texas. If you find a hognose snake anywhere in the hill country, it's an eastern.
Very good. Did you show contact information on any of your slides? You've got a question about.
Oh, I didn't, but I'm very easy to find in a Google search.
Yeah, and I know the person who sent that, so I can contact them. Somebody asked a question: if we find a hognose, should we report it? If so, where?
No, it would be valuable if you're a person who enjoys iNaturalist to put it on iNaturalist and link it to the Herps of Texas project. But outside of that, you don't need to report it. I will say, I don't ever hope to find a result. I would have been happy with saying that, hey, we have two species or not. I will say this: if there had been two species, if we could say Mexican hognose are truly distinct, I think the South Texas populations would have warranted listing for protection, but I don't, there's one species, it's the same thing. And that animal occurs, this is obviously outside the state of Texas, occurs in New Mexico and Arizona as well as Mexico. And they're doing well in several places in Mexico. So not good for our Texas animals, but they're still doing well.
Excellent. Couple of more questions coming in here as we have to wrap up here in a few minutes. But
yeah, I saw Dave's in here. We're going to publish this in a herp journal, and then we have the hognose genome as well. And I don't know where we're putting that.
Fantastic. Somebody asked about the presentation. Yeah, we're gonna be posting the presentation to YouTube, so you can check it out. We have a playlist on our Parks and Wildlife Education channel, so you can get access to that. And the last question I can see on here is about feral cats. You want to speculate on that at all?
They eat hognose. Actually, there was an animal in that presentation that was caught by a cat, and it survived.
Okay, very interesting. Well, that appears to be the end of the questions.
Thank you for coming and enduring the technical difficulties.
Yeah, but these things happen, don't they? So no, it is all good. I'm just going to wrap up here. Corey, thank you again for joining us. It was a great presentation, really interesting work. Looking forward to seeing